think it's live. So good morning, afternoon, and evening, everyone. Welcome to this session on open data with novel techniques. We have 4 exciting speakers and talks lined up. So I think without further ado, I can introduce our first speaker, Chaoyue Wang from the SJTU Ruijin UIH Institute for Medical Imaging Technology from Shanghai, China. He'll be speaking to us about quantitative susceptibility mapping in the UK Biobank brain imaging.
Oh, I can share my screen.
Yes, I think so.
Okay. Okay, let me share my screen.
Yep.
Cool. Okay.
Perfect.
So first
Take
of
it
all,
away.
I'd like to thank the committee for the invitation and for organizing this amazing workshop. My name is Chaoyue Wang. Today I'm going to talk about our work on quantitative susceptibility mapping in UK Biobank brain imaging, which offers one of the largest open data resource for neuroimaging. This work was done when I was working in Oxford, but I've recently started a new position in Shanghai, China. So first, I'll start off with an overview of the UK Biobank imaging study, which is the foundation of this presentation. So the UK Biobank imaging study is aiming to scan 100,000 subjects with a multi-organ protocol. Currently, data from more than 40,000 subjects are available, and these subjects have undergone extensive baseline measurements, including environment, lifestyle, and genetics in the larger UK Biobank project. Subjects are largely healthy when scanned, but they may develop a range of health issues over the coming years. For that reason, a particularly valuable feature of Biobank is that we have long-term access to subjects' health records. As shown on the right-hand side, multiple modalities were included in the brain imaging protocol. My work has been focusing on the susceptibility-weighted MRI data, in particular to do quantitative susceptibility mapping, or QSM, sorry, can you see my, not sure.
It's minimized. Yeah, now it's back.
Okay, which is what I'll focus on in this presentation. Sorry about that. Okay, so how do we get a sense of the scale of the numbers of subjects that will be scanned in UK Biobank? So here I will use a video to give you a visualization of the size comparison to other imaging studies. And so a typical imaging study commonly has a few hundred subjects. If a study has a few thousand subjects, then it is considered big. And here comes the UK Biobank cohort with the ultimate 100,000 subjects. And we will have QSM data available for all these subjects, which gives us a data resource that is at least 2 orders of magnitude greater than any existing QSM datasets. And given the age range of UK Biobank subjects, in a few years after the imaging scan, many subjects might develop a range of neurological disorders, including Alzheimer's and other sorts of disorders. And in addition, UK Biobank is particularly powerful as a result of the combination of the number of subjects and the breadth of linked data. As shown here, subjects have been extensively characterized and consent to allow access to their long-term health records. So with UK Biobank, we could learn the relationships between various biological measures and health outcomes using technologies such as machine learning, and to enable prediction of subjects' long-term health outcomes from all these biological measures. So this is a quick overview of the UK Biobank imaging study. And next, I'll focus on our work that used the susceptibility-weighted data for QSM. So the first question is, what is QSM? So QSM is a technique that generates a quantitative voxel-wise estimate of the underlying magnetic susceptibility of tissue. And the map looks like this. And it is particularly powerful as a number of tissue constituents associated with disease have distinctive magnetic stability properties, including myelin and iron, as shown here. So this susceptibility-weighted MRI in UK Biobank is acquired using a gradient echo or GIU sequence to achieve a short scan time of 2.5 minutes. UK Biobank decided to compromise slice thickness because the primary goal of this acquisition was to image microbleeds. But it's worth noting that 3 millimeter as a slice thickness is not ideal for QSM, but we can still do QSM using this data. Nevertheless, they collected both magnitude and raw phase data, which enables us to do multiple things with this data. But here, I'll focus on the quantitative side, or images that can be derived from this acquisition. So first, we can generate quantitative T2 star maps using the magnitude data, which is a very straightforward processing and relatively straightforward to do. So UK Biobank has already produced T2 star maps. My work was to process the phase data for QSM. Although T2 star QSM are derived from the same GRE data, they contribute complementary information. For example, in neurodegeneration, we often see myelin loss, which will lead to increase in T2 star and susceptibility measures. We might also see greater iron deposition in tissue, which will lead to decrease in T2 star, but increase in QSM measures. So we really need both QSM and T2 star when they are coexisting changes in myelin and iron, for instance. To do QSM, a multi-step processing of the phase data is required. Here I'm showing the QSM processing pipeline we have developed for the UK Biobank data. You don't need to read this pipeline. I just want to highlight that there are multiple algorithms available for each processing step, indicated by the blue arrow. And we have performed extensive evaluations of these algorithms and selected the optimal combination of algorithms for this specific data set. So this pipeline was used to process the data from the recent release of more than 40,000 subjects. And given that it produced voxelized maps, so the QSM data for all of these subjects will be huge. So that's why in UK Biobank, we need to reduce the amount of data that we need to work with. And we commonly produce summary measures that are sets of scalar values extracted from each subject's imaging data. And these summary measures are called imaging-derived phenotypes, or IDP. So for QSM, we produced two sets of IDPs, or imaging phenotypes. The first set is targeting the subcortical regions. We calculated the median susceptibility in 8 subcortical regions shown here. And another set is QSM white matter hyperintensity IDP, calculated as the susceptibility difference between hyperintensity lesions and normal white matter. So UK Biobank has also produced T2 star IDPs, calculated in the same way as these QSM IDPs, which enables comparison between the QSM and T2 star estimates. So after reducing the data size and we have this imaging phenotypes, that we were able to reduce data size to a few numbers per subject, which makes it much easier to perform analysis, such as associations with genetic and phenotypic data. So next, I'm going to show some example results from genetic associations. So in UK Biobank, we have genetic data consisting of over 20 million genetic variants per subject. A genetic variant is a site in DNA that varies across subjects. And we also have a QSM IDP. We also have QSM IDPs from the same subjects, so we can study the cross-subject correlations between them. And we've done this correlation analysis for the whole genome, which is called a genome-wide association study, or GWAS. So here I'm showing the GWAS result for a single IDP, medium susceptibility in right thalamus, using what is called a Manhattan plot, where each point represents one genetic variant, and x-axis gives its location on the genome, and the y-axis gives the significance of its correlation. Two horizontal gray lines indicate the standard significance threshold and after multiple comparison correction using Bonferroni methods. Any genetic variants that passed threshold were color-coded in black. We see that significant associations are clustered together, such as here in chromosome 4. This is expected due to an effect called linkage disequilibrium, or LD, in short. So here we only focus on the top variant, which is called a peak association. And these genetic variants have their own ID, unique ID, that we can use to look for related genes. And in this study, we identified genes related to a broad range of biological functions, including iron, calcium homeostasis, myelin or glia, brain development, and general regulations, including regulations of gene expressions or cell functions. So this is just a result for a single IDP. And if we sum up all 16 QSM IDPs, in total, we identified 229 peak associations with these QSM IDPs. As a comparison, 225 peak associations were identified with T2 star IDPs. So here I'm showing you the summary result for QSM GWAS with all ROIs, where the y-axis represents each ROI, and the x-axis gives the number of genes associated with a given ROI. We can see iron-related genes in red are associated with every ROI, and the 4 ROIs, substantia nigra, palladium, putamen, and caudate, have the largest amount of genes correlated. And these 4 genes also have highest amounts of iron among these ROIs, or even in the brain. So producing IDPs is an efficient way to reduce data size and discover new associations, but it loses rich spatial information in voxel-wise maps. So after identifying genetic variants of interest, we can further explore the spatial pattern of association. Once QSM maps are aligned to a common coordinate system, we can correlate the genetic variant of interest with susceptibility values at each voxel location to perform a voxel-wise correlation. Here I'm showing the correlation R value map for a variant related to gene HFE, which is related to iron absorption. R values are plotted as color overlay. Yellow, red indicates positive R, and blue indicates negative R. We can see the associations are mostly in subcortical regions, including putamen, subregions of the thalamus and substantia nigra, but also in regions that are not captured by our RI, such as the red nucleus and regions in the cerebellum. And most of these associations are positive. And if we look at another variant related to a gene that is related to iron export, the first thing to notice is that most of the associations are now negative, but the spatial pattern is also different. For example, this variant is associated more with palladium, not instead of putamen. So we also identified associations outside subcortical structures. For example, a variant related to gene GFAP, which might sound familiar to some of you. It encodes a glia protein, and the associations are now mostly in white matter. So here, so these are the results that we derived from the genetic association analysis. And besides association analysis, we have also performed the estimation of heritability of QSM IDPs. So heritability calculates the fraction of IDP variants explained by a linear combination of all these genetic variants. So in this plot, horizontal axis gives the value of the heritability estimates, and the vertical axis again represents subcortical ROIs. The circle represents the heritability estimates with arrow bars at 2 sides. Here, the highest heritability estimate is from left putamen, and it is roughly half the heritability of human height. So these are just some example results that we have derived using the combination of genetics and QSM data that are offered in UK Biobank. Next, I'm going to show again some results using association analysis with phenotypic data. And as I mentioned, in UK Biobank, subjects have also been extensively phenotyped, which offers us to the opportunity to explore phenotypic associations, again with all kinds of imaging phenotypes. In UK Biobank, the non-imaging phenotype consists of over 17,000 variables. We have grouped them into 17 categories, including lifestyle, blood acids, and health outcomes. Similar to GWAS, we can correlate these phenotypes with our QSM IDPs. In total, we identified significant associations between 251 phenotypes with the QSM IDPs, and as a comparison, 224 phenotypes are associated with G2-star IDPs. Again, here I'm using a Manhattan plot to show the phenotypic associations between a few phenotype categories and subcortical IDPs. Here, each circle represents one phenotype, and different color indicates different IDPs, or in other words, different subcortical regions. The x-axis indicates which category a phenotype belongs to, and the y-axis gives the significance of its correlation. The horizontal dashed line represents the Bonferroni-corrected significance threshold. Given the large number of association axes, I will only highlight a few associations of interest. As aforementioned, iron is one of the major sources of contrast in QSM, and we identified many genetic variants related to iron homeostasis. So not surprisingly, the strongest association across all phenotypes is with hemoglobin, which is a marker for body iron level. And this association is positive. And we also identified the positive associations with smoking status and alcohol intake frequency, which are both known risk factors for a range of diseases. So these are a few phenotypes with possible links to magnetic susceptibility, but there are many other phenotypes that do not seem to have these direct links to susceptibility measures, which definitely needs further exploration. Given the time, I'll skip this one. So basically, this plot shows the association result with QSM wet-matter hyperintensity IDPs. We can see in health outcome category, the strongest correlations are with multiple sclerosis, which will have white-matter hyperintensity lesions in patients, again, as expected. So in conclusion, UK Biobank will produce QSM data resource that is 2 orders of magnitude greater than any existing QSM studies. And we find that stability measures in the brain are associated with many genetic variants, phenotypes, and health outcomes. And the QSM reduced do offer complementary information, and our data will be made available to researchers worldwide via UK Biobank. So together with other imaging data, UK Biobank will be a powerful open data resource for all kinds of studies. And if you'd like to see more results, I have a link to our paper down below. And I'd like to thank all the contributors and our funding for this work, and thank you for your attention.
Thank you, Chaoyue, for the excellent presentation. I would now like to invite the second speaker, after which we have a 10-minute discussion period. So I'd like to now invite Steffen Bollman from the University of Queensland in Australia, and he'll be speaking to us about open and reproducible data using the Neurodesk platform. Steffen, the stage is yours.
Yeah, thank you very much for the kind introduction, and welcome everyone. So yes, I'm a research fellow at the University of Queensland, and today I want to show how we can use the open source Neurodesk platform to enhance open data workflows and make neuroscience software more accessible and making analyses more reproducible. To start, I would like to acknowledge the country and the traditional owners of the land on which we're meeting today. For me, here in St. Lucia, these are the Turrbal and the Yagara people, and I pay respect to their ancestors and their descendants. And another declaration I would like to make is of potential conflict of interest that could be perceived with this presentation. So there are two, I receive research funding from two companies. One of them is Oracle for Research, and Oracle for Research partially funds the Neurodesk project that I chose today via cloud credits. And I will also talk about a project from Oracle for Research, which I'm involved with, and I work with Oracle Research on this project. It's called Oracle Open Data. And I also receive research income from Siemens Healthineers, but there's nothing related to this presentation today. Then I would like to start with introducing an open data example, because I think it's always nice to start with a cool example. And then that example will take us through this whole presentation, because then I will show the Neurodesk platform and the open data workflow. Then publishing open data, how can we get our data out there? Then accessing open data, how can we use open data? And in the end, I want to do a little excursion into re-executable papers that link code and open data together. And we'll look at how close are we to this and what can we do with this. So I want to start with a statement. Open data is easy, right? It's simple. We just zip up the whole directory. We upload it to Dropbox and we dump the link in the paper, right? I mean, this is open data, right? Well, wait a second. The reader now has to download a multiple gigabyte file just to see which functions were used. That's maybe not great. And then also, will the code still work in the next version, for example, of MATLAB? Does the reader even have a MATLAB license? What if this free service stops? What if Dropbox gets bought by a billionaire and they just reform it to stop public links? Or I found a bug and I need to update this thing. What do? Do I just replace that file and just hope no one notices? Yeah, so I think it's maybe not ideal yet. So we thought about how could we do this better? And this is from a recent paper from our group. We thought maybe we can even provide like an interactive version of our implementation. So something that runs in a browser where a researcher doesn't have to do any setup work to get it to run. They can just click and they can see what we've done in the paper, and they can see the results and reproduce the results. And yeah, we need a cloud-based platform for this. And in this example, we used Google Colab. But what if Google stops one of these free products, right? Google has been known for this. So here in the paper, we actually don't write that we use Google Colab, because we would be able to change that platform later on. So I will show what enables us to do this in a second. So we also said, okay, let's not mix the source code and the data. Let's actually keep these things separate. Let's put the source code in an easily accessible way on GitHub. And also, let's say this commit on GitHub was actually used for the paper. But the cool thing is now we can actually continue. We can incorporate bug fixes and actually change these things. And it's very transparent what code was used for the paper, and then also that the code can evolve past the paper. And this is this link that allows us to change all the things and keep flexible with this in case, for example, this cloud-based platform goes down. It's the Open Science Framework. And here we can update the links and the data if bugs are found or services move on. So now let me start with the Neurodesk platform and the open data workflow. So what is Neurodesk and which problems are we trying to solve? So Neurodesk is an open source platform for accessible and reproducible neuroimaging. And our aim is to make neuroimaging more accessible by tackling these three big challenges here. So the first one is that research software is difficult to install. So you have dependency conflicts, you have a lack of packages. You can't just simply apt install FreeSurfer, usually. Also, there is a lack of maintenance because researchers move on to new positions and then packages don't get maintained, but they're still crucial for our work and we need to be able to reproduce old results. So there are a bit of challenges around in this space. And then also we work with variable compute environments and operating systems. So we use Windows, Mac, and Linux. We use high performance computing systems. We use workstations in the lab. We use our laptop. We use cloud computing. We use Jupyter notebooks. And if we want to analyze data in all of these environments, it usually means we have to bring the data and the software there, and we need to start all over again. So to me, it frustrated me a lot in the past years that I always had to do this and always costed me a lot of time to set up my environments. Another big challenge is large data sets. So it's not that easy to simply take a 20 terabyte data set, like the UK Biobank that we just saw, and just download it on your computer, right? Your computer will run out of disk space. So it's actually not that easy to handle these things. So it would be really cool if we can bring our analysis packages to the data, and we can be really flexible in this. And that's what we built. So I show here the funders. So we're mainly funded by the Australian Research Data Commons to build this project, by the National Imaging Facility, but also Oracle for Research, as I said, and two universities, Swinburne and the University of Queensland. So it's a well-funded project, and we're working on this since 2 years now, and we're in the last year of this project. So what did we build? So the most important thing for us is the community. So Neurodesk is centered around the neuroimaging community. And the community builds and maintains software recipes for neuroimaging workflows. And the advantage is that developers of these tools can contribute their software tools very easily. They simply need to provide us with instructions on how to install their software. And this is then turned into a recipe for building a so-called software container. I will explain what this is in a second. So, but in the end, and we see this here, in the end, the community uses all these things again. And now the cool thing is that the whole community profits from all the domain expertise of everyone out there. And basically everyone maintains the tool they're most familiar with, and then everyone can access tools that are actually working. So this is our core principle. Now, these software recipes contributed by the community, they are automatically built and turned into software containers, and then they are distributed. So importantly, every tool and every version is available as a separate container. And therefore, and you see this here, like we have FreeSurfer 711, FreeSurfer 720. With this, we avoid dependency conflicts between tools. So this means we can install FreeSurfer 711 and FreeSurfer 720 next to each other, which you can't do on a normal computer easily, because there are dependencies that these things depend on, and they need to be updated. So sometimes, like an older version of FSL, you cannot install correctly and run correctly on a new operating system because, for example, you miss libpng, which was deprecated in an older Ubuntu version. So it's very, very tricky to run old software, for example. And also, it's tricky to run new software on old operating systems. So let's say you run an HPC, an high-performance computing cluster. They often run very old versions of Linux because it's a pain to update these things. So containers really allow us to package everything we need to run the software. And we see this here, like we have binaries and libs. So these are like libpng and all these fundamental libraries that a software needs, plus all the binaries that a software needs, plus all the dependencies, conda, Python, MNE, every package that this software needs, we can package in a container, and then we can move this between operating systems. So with this now, we're not dependent anymore on the operating system. So we can take the software and run exactly the same thing on a high-performance computing system, on Linux, on Mac, on Windows, and it behaves identically everywhere. And this is really cool. We also build an accessibility layer, and you see this little thing gray down here, and it's almost a little bit undervalued. What it does is, I'll quickly explain it. So basically what we've done is we try to hide all the complexity of containers, because running containers and building containers is really a pain, and you need long commands, and it's not fun. So what we did is we just hide that it's actually all running in containers, and we make it really easy to access and run the software. And we take care of all the complexity of getting the container to you. For example, you don't have to download these containers. So for example, FreeSurfer is a 10 gigabyte container. But if you download it through a low internet bandwidth connection, the download would probably stop. And we have these problems in Australia. Like we have people with low internet bandwidth, and they never, in the beginning of the project, we had these big containers. They never managed to download them completely. So what we did is we use a software system called CVMFS, which was developed at CERN. And think about it as Netflix for software. So basically, it streams software to the end computer, and it works through a very low bandwidth connection. And the cool thing is people don't have to wait until these things are downloaded. They can click on FreeSurfer, and it just opens up. So with this, we can make multiple 100 gigabytes of software available on pretty much any system out there. So now, finally, we have various ways of interacting with these containers. As I said, we made it very transparent how these containers work. So for the user, we have a graphical interface, which is called Neurodesktop. It's basically a full Linux desktop accessible via the web browser, and it runs in Linux, Windows, and Mac OS. And we built a command line tool. as well that enables us to use these containers on, for example, high-performance computing systems in Jupyter notebooks. So really with this, we have the full flexibility of running the software where our data is, where we need it, behind hospital firewalls, wherever it's required. So this setup really now allows us to bring the analysis environment to the data and brings it closely together. So now we have easy access to the software and we can process large amounts of data, which is really, really cool. Okay, here's just a little example to show this accessibility. And we incorporated, for example, Neurodesk inside Jupyter Lab. And I'll share links here in the chat. So you can actually explore this yourself. So basically, we set this up for BrainHack, which is last week and this week. And basically choose the region that's closest to you. So the first one is for North and South America. The second one, BrainHack Sydney, is for Asia Pacific, and BrainHack Europe is for Europe and Africa. So basically think of it as the three big regions in the world. You want to pick something that's close to you just for latency reasons, but every server will work for you. And we could also easily launch servers in other regions of the world, but this is just where the BrainHack events were this year, and we just launched it close to where all the users were. And now the cool thing is you can click on this little button in there, Neuro Desktop, and then it brings up a full desktop in the browser. It has all the tools installed that you can think of. These containers solve all these dependency issues. We have full GUI support, so all the graphical interface works. And it comes with all the tools required for curating, processing, creating, and using open data. So it's really the centerpiece of an open data workflow. So now I said this works. So how does it work? Publishing open data. Where can we store data? Which tools exist to get our data openly available? And here I want to show one particular service that I've used quite a bit in our papers, because I think it's a very, very well-designed service. It's called the Open Science Framework. It's supported by the Center for Open Science. And this creates this really cool link, osf.io, which I can put in a paper and I can update things later on. So why does this work? Okay, so the Open Science Framework is an online platform that enables researchers to plan, collect, analyze, and share their work. And it's a non-profit organization called the Center for Open Science. So they got funding from a couple of sources to build this center. And the cool thing is, even if they run out of funding, they have a preservation fund. So that means that the data I deposit there is 50 years plus read access. So basically, for the rest of my life, I can be sure that my data will be accessible from people who click on the link in the paper, and I can also update it. So that was, for me, the reason why I'm using the Center for Open Science for this. And then it's really cool because you can connect various services in the Open Science Framework. So here I just connected my GitHub, I connected OwnCloud. So you can use internal OSF storage to deposit your data, but you can also bring institutional storage. So let's say your university has OneDrive, you can just connect OneDrive, and then you can link your data that you have on OneDrive into the Open Science Framework. And now the cool thing is that once you are happy with everything, you can register your study. You can say this thing was published. And then what OSF does is amazing. They take the data from your OneDrive, put it in an archive, give you a digital object identifier, and they guarantee you that this data will be available for the next 50 plus years. And I think this is really, really cool. And it does this with all the services you can link. So this means you can really preserve the state of your paper, of your data, of your code at the time of publishing. So now, how do you get my data there? So there is a really cool tool, which is an open source tool. It's not officially supported by the OSF framework, but it's called the OSF client. It's just an open source framework that allows you to do an OSF init in a directory. Then you get a project, you authenticate to the OSF, and then you can do OSF upload -r of the data you want there. And then it just uploads it to the OSF, and everyone can access the data once you make it public. So pretty nice and straightforward. And then you get such a website where you can change everything you want. You get the data that you uploaded there. You see the history of what happened with the project. And yeah, it can live after the paper was published. So now I want to show another way of getting data up to the OSF. If you don't want to use the OSF client, there is something more powerful that you can use. And the project is an open source project, again, data-led. It's a free and open source distributed data management system. And it does a lot of things. It's super, super powerful. But one thing why I want to highlight it here is it integrates with including the OSF. So with lots of data infrastructure, and it can upload to the OSF. But in a nutshell, what it does is it's Git for data. It uses Git and Git Annex under the hood to do this. But basically, you can version control your data similar to how you can version control your code. But because with a normal Git, you can't easily version your data because Git would just fall over. It's not made for that. But DataLet is made for data. So you can version multiple terabytes of data without any issue. And this is how you can create a DataLet data set. And then you can just add your files in there and run DataLet save with a commit message. So you see it's very similar to Git. They try to keep it all nicely parallel to Git workflows. And then it just adds these files and it keeps track of what happens to these files. Now, you can authenticate then DataLet to OSF with DataLet OSF credentials. And then you can create a sibling where you say, now I want to share my data, and you can upload it to the Open Science Framework. And then everyone can access your data using DataLet, which is really nice. So now we got our data shared. Now we want to efficiently access the data. So do we have to download everything at once? Because let's say we have a 20 terabyte data set. That's not really cool, right? So let's see what we can do there. So here in our paper example, we wanted to build an interactive Jupyter notebook to just make it very easy to explore the data and quickly change things and really build on what we build in a paper, because ultimately this is what we want to do in science, right? We want to step-by-step progress, and we want to enable someone else to take what we did and build it further and develop it further. So here on this website, on the OSF, we just said the interactive computation notebook can be found here. So the cool thing is, currently this is running on Google Colab. So when you click on this, you get the Google Colab notebook, because we needed GPUs for this, and Google Colab still offers free GPUs. It's a deep learning application, so we can nicely show how the data gets into the framework, how it gets trained, how it gets predicted, and people can really reproduce the results from the paper and then quickly change it and do their own application. So that's quite cool. But if, for example, Google Colab would disappear, it's not a problem. We can update this link anytime. So installing the OSF client in a Jupyter notebook is straightforward. Pip install OSF client. It just doesn't need much. It really installs pretty much everywhere. And then you can just do an OSF-P project ID clone, and then it downloads the data that you have into your notebook environment, and then you can explore it and you can do whatever you need to do. DataLet can do the same thing. So if you published your data with DataLet, you can do DataLet clone from OSF. And then there is a really cool feature of DataLet. It doesn't download everything at once. So it's like Git. It clones the repository, but actually it doesn't download all the data. So this means you can clone a 20 terabyte data collection, but then you can say, I only want this one file. And then with DataLet get of the file or of a directory, it then actually downloads the data you want. So this really gives you a cool way of working with massive amounts of data. So that's why I wanted to highlight this tool, because it's very, very useful for exactly this use case. And then I showed DataLet here in connection with the Open Science Framework, but of course it works with lots of other data providers. I specifically want to highlight OpenNeuro, which has lots of functional magnetic resonance imaging data sets in our field, and all the other providers there, which really provide amazing open source data. So then I want to show another service, and I mentioned this in the beginning, that I'm working together with Oracle on a platform that we call Oracle Open Data. And what they want to do there is they basically provide free open data hosting. So you can apply and you can say, I would like to have my data set hosted on Oracle Open Data. And then you get listed in this directory. You can upload your data. And we did this with one of our 7 Tesla MRI data sets. We scanned multiple people over multiple years. So it's a longitudinal data set, quite high resolution for hippocampus studies mainly, and for segmentation studies. So we uploaded it there. And then the cool thing is Oracle Open Data makes it quite straightforward to use this data. So you can just, you see that here, you can curl or wget the data in Linux, or just download via HTTP. But that's not why it's cool. That's just standard. But why is it so cool? You can, in Neurodesktop, or pretty much in any Linux system, you can use a system, a file system that's called S3FS. So S3 is the object storage of cloud native storage. And what you can do is you can mount this data set directly into your compute instance. So now the cool thing is, once you run this command, you get a little, you can just open it in your browser. You can just click through the files, and you can just double click on any file and just directly work with anything that you see there. And it's really straightforward then to build on this. You don't need to complicate download or anything. You can just quickly explore it, and it's really nice, and it does take care of everything in the background. So it nicely integrates compute and open data into one framework. And in Neurodesk, we made all of these things very easy. So the OSF client, data-led, this S3FS with Oracle Open Data, all of this is nicely integrated and directly available for everyone to use. So now I want to come to my last little section, which is how close are we really to a re-executable paper, and can we use neuroimaging software inside Jupyter notebooks? And I just want to highlight two initiatives there. There are more, but I just wanted to pick two really great ones from our field. So there is the Neurolibre project, which I really like. It uses basically computational notebooks to build a re-executable paper in our field, and that's really, really nice. There's also eLife. eLife has also like an interactive paper where you can, similar to Jupyter notebooks, build with R Markdown and Jupyter notebooks an interactive figure, and then people can change your code to the figure and explore how you got, for example, to your figure. So now, what we can now theoretically do with Neurodesk is we can use now all neuroimaging applications inside computational notebooks. And I show this here. So we integrate with LMOD, and LMOD, if you use HPCs, you know that's a module load command. So inside Python, we can just say import LMOD and then load FSL. And then I can use, for example, BET from FSL 6.04. So I can really control the version. And then I can just use, for example, NiPipe and write my pipeline, share it with anyone. And then I could really build a whole analysis of my paper, re-executable. So I hope that we can get this integrated in these other projects that use the computational notebooks already. And yeah, really integrate our neuroimaging software, the rich ecosystem we have into these re-executable papers. And yeah, here I just wanted to highlight these are the modules, so you can also just click on them and say load that module and then launch a new notebook, and then the executables are available inside the notebook. So it looks like magic, but in fact it's just hard work. So if you explore it, and yeah, let me know if you like that, what you would like to see there. So as I said, it's all an open source project. We are super happy to get input on these things. So that's, for example, just an outcome of last week's brain hack. So it's a very new feature. So with this, I come to my end, and I hope I could show that the Neurodesk platform makes it really easy to access, process, share open data. We can really nicely publish our data, share re-executable workflows, and make it nice and easy for other people to build on our work and take our work further. I thank you very much for your attention. If you want to see more from our group, we have 4 posters in the poster hall from Ashley, Tui, Fernanda, and Corbinian. There's 3 postdocs in my group and 1 PhD student, and we're all working in the space of reproducibility and pipelines. So if you're interested there, definitely look at their papers, look at their posters, and yeah, really get in touch and get and join the Neurodesk project. As I said, we're very open for contributors, and yeah, would love to see you out there, and I'm looking forward to the discussion. Thank you.
Thank you, Steffen. I know it's the afternoon on the 7th, so this talk was really futuristic, and I really do think it's the future of neuroimaging research, especially given all the innovations that you and all the collaborators have worked on. Now we enter the discussion section, so I think Chaoyue could join us.
Yeah.
Hey, yeah, so I do see 2 questions already in chat. Okay. The first one is congratulating you about the impressive work. With so many data sets and so many associations, what are the main challenges with working with this huge data set and deciding what to focus on?
Cool, thanks. Yeah, that's a good question. So the challenges are, obviously, that's a lot of data at the level of PB. It's not terabytes, it's PB level. So the first challenge is we need a very established data storage system to handle these large amount of data, and also to analyze it we need a very powerful computing platform to enable all these kind of analysis. Actually, in Oxford, we have a very strong computing platform to do all these, to enable us to do whatever it takes to process these data. For my specific work on QSM, there's the challenge would be, as I mentioned in my presentation, QSM requires a pipeline that post-process the face data, and there's various steps involved. And because we have such a large cohort, we can't inspect every result from each subject. So we have to really have a very stable and automated pipeline to do the QSM processing. So that's why we spent quite a lot of time evaluating the existing toolboxes and algorithms to find the optimized or the best combination of algorithms that will be optimized for this specific data set, because there are interactions between the image parameters and data quality with the choice of processing methods. So by doing all those kind of evaluations, we are able to develop or find a pipeline that is stable. We don't see much artifacts at the QSM maps, and it is fully automated. So we can just with click of a button or just press enter, then we can process all these 40,000 subjects. So that's kind of like the challenge we have. And then for the second part, and that's a great one. We have, as I showed, there's in the Manhattan plots, there are lots of associations, and they are in different categories, different gene biological functions. So for our work, the goal for our work is actually to produce this data resource. So because Biobank has already collected these GRE data sets, but they haven't been processed for QSM. So my work was to do the processing, and we produce these QSM maps, and we extracted several summary measures, or what we call IDPs, that are just representative measures that one can take from QSM maps. But there are lots of other measures that they can do with these QSM maps. That's why we call this an open data resource, because these QSM maps have already been released via Biobank. So researchers can apply for access. Then they will have QSM maps for these 40,000 subjects. Then they can explore their associations or regions of interest. But for our work, it's more like we just need to demonstrate the power of this data resource. It's in our paper. So we just selected a few representative regions to report, and also interesting and also associations. Some are new, but some are also validating previous findings. But the most of the remaining works will be done by researchers worldwide. Hopefully, there will be lots of groups taking up this data resource and do their follow-up, all kinds of.
I think it actually you answered one of the other questions, which was, can you comment on data availability and if QSM data, the raw magnitude and phase, will be available to the public?
Yes,
so it's released already, or
the voxel-wise QSM maps are released. But if we don't already have access to UK Biobank imaging data, then you have to apply for that. An average application time is around 20 weeks. Wow. So it's not a fast process. So we need to apply for the access. Then we download the data, and the voxelized maps are available, I think. But IDPs are not, surprisingly. IDPs are just a bunch of numbers. But once we have voxelized maps, you can take the median by ourselves. But the data are available, both magnitude and raw phase data, and the process T2 star maps and QSM maps. they are both available now.
I just have one follow-up. So about how you chose parameters for the QSM. I know from my limited experience that it can be notoriously different depending on what numbers you choose and which algorithms you choose. Did you have any QC criteria that you used to assess what was good compared to the others?
Yeah, thanks. Yeah, that's a good question. At the stage of acquisition, because as I mentioned in my presentation, it was the CRE acquisition was originally for microbreeds, so they have very thick slices. But for the purpose of QSM, it's not an ideal parameters to start with. So we have very thick slices, so it's anisotropic voxels, highly anisotropic, and then it collects its raw phase data, so we can do QSM processing. And as you mentioned, there are lots of algorithms available and also parameters. So we have a QC or evaluation pipeline to test. We have tested more than 25 even 30 combinations of different algorithms for different stage. We test different available softwares, and also they are all well established. So there are lots of people are already using these kind of algorithms and methods, toolboxes. So we just have evaluation pipeline. We tested many combinations of different algorithms, and also fine-tuning the parameters, even contacting the development team of specific toolboxes to get feel of the what will be the optimized parameters. And
it was scored by somewhat like assessors, or like it was judged by raters. Like how did making the decision between it was?
Yeah, that's a great question. So I had a slide that I showed different combinations of the algorithms, the effect of that on the final QSM maps, average through 1,000 subjects, for instance, and we can see that there are dramatic differences with the same set of subjects, but just processed using different combination of algorithms. The QSM maps averaged, they look quite different. So that is the problem with QSM in general. That because we're solving an AO-post problem, so people have been taking different strategies to overcome this problem, and resulting in a for some algorithms there may be more, and we don't have a ground truth, right? So we are more relying on the visual inspections, whether there are a number of striking artifacts, and just in general, whether the QSM maps are homogeneous or not, without any obvious artifacts. That's for visual inspection. But we also did some quantitative evaluations, basically measuring, for instance, medium in subcortical regions, and plot that across a few thousand subjects to see the variability. But again, we don't have ground truth. We don't know whether they should have a large variability, like variance across these set of subjects, or we should have a more homogeneous or more tight distributions within the subject. So it was a tough decision to make, but I think based on the mostly the visual inspections of the artifacts and whether the maps are homogeneous and output from each processing stage, we were able to identify the what we thought was the the best combination.
That makes sense.
But again, UK Biobank is releasing raw data, so
people can process it however they want, right?
We're just demonstrating that this QSM is possible, and we can identify lots of associations, and they are very interesting. And
I do see some increased votes in the chat, Steffen. So it's from Julien, Cohen-Adad. Firstly, complimenting you about the very nice overview of the present and future of neuroimaging. So the one question about data sets that are open but require a private token to be downloadable. Example, the Courtois Neuromod dataset, which you listed in your presentation. how compatible is Neurodesk with such data sets?
Yeah, thank you, Julien. That's a great question. So yes, it's fully compatible with this. So all the tools that I showed support token authentication. So the OSF command line client can authenticate to a private project. So that's how you upload it in the first place, and then also download and test everything. And then when you switch it public, you don't need the token to authenticate anymore. DataLet is the same. DataLet supports token authentication, and exactly, lots of these data sets, they require a sign-up process where you need to sign ethics or something, and then it supports entering a token. And the same is true for the S3FS approach that I showed. I showed a public data set that doesn't need a token, but you can just include a token in the command line that's then required for authentication. And with this, you can access all data sets really. So it's it's fully built in, because it's a crucial aspect. Yes, but
yeah, I think you also answered like one of the other questions, which is, can storage accessed via S3FS be registered in OSF as well?
Exactly. So potentially it can be. I haven't tested that, Oren. S3FS just requires a public bucket. I just checked because I saw your question. The files are on object storage on OSF. I need to talk to them if we can access them without authentication, or if we need a specific key to access them. But it's something we could easily do. Yeah. Cool. And
I had one general question before we get to the top voted one. So it's more about like how, in your experience, Steffen, has it been to encourage traditional neuroimaging labs to adopt open data processing and an open data culture? Has it been tricky, challenging, or do you find many receptive to it?
Yes, exactly. So what I see currently with the adoption of Neurodesk is that people would love to do open science and publish data sets openly, but then they get to the point where, okay, I don't know how to do it. And then people say, oh, just download this tool, and then it works. And then they try to install the tool with pip, and it doesn't work. And then most of them just give up frustrated, right? And being able to not upload it. So what we see with Neurodesk is, by making this a lot easier, people are very happy. They just use it because it doesn't require any extra work. They don't need to install anything. So I think if we make it very easy and make it the default, then this will become a lot more common. So I would say most people would love to share their data. Because they get more public, more citations, right? It just makes a lot of sense. And yeah, by making it easier and better, then we get more adoption of this.
And the other question was, well, again, compliments for an absolutely amazing presentation. What are your thoughts on research software engineering as a different but parallel field to academia?
Yes, exactly. I wish our research software engineer would be here now. So the project that Neurodesk is built in a community project. So they have 3 big universities, Swinburne, Sydney, and Queensland. And in Queensland, we have the luck that we have Ashwin Narayanan on the team. He is a research software engineer. He works for the National Imaging Facility to look after all our imaging software and repositories. And yeah, he really has that career path. And I think it's super, super crucial to have people like Ashwin. So without him, this project would not be anywhere, really, because he really built a lot of the fundamental aspects behind the platform, all the server infrastructure, a lot of the GitHub, GitOps things. That's all he built that. And I'm a researcher. I don't know a lot about these things, so I ask him every day how things work. So I think it's super, super crucial to have people like Ashwin, for example, on the team. Funding people like this is indeed not always easy. So in Australia, we are quite lucky, I would say. We have like a couple of infrastructure funding schemes, I would say. Like we have the Big Entry scheme, which is an infrastructure provider for Australian imaging and all infrastructure, all research infrastructure, but imaging is a big pillar. We have the Australia Research Data Commons, which funds projects like this, which funds Neurodesk. And for example, also in my team, we paid a postdoc to work in a more software engineering role. But having said that, a lot of the contributors to the project do this in their free time. They're just normal researchers, whatever that means. But yeah, contribute in their free time. And what we get out of it from it, we're currently writing a paper about the project. So basically, people get a co-authorship. But I think currently it's very difficult for an academic promotion to say, for example, hey, I'm running an open source project and it has like 1,000 stars on GitHub and it's really used all over the world. Unfortunately, no one cares. People in my promotion committee just look at the H factor that I produce and how much grant income I get. So I think this makes these projects very tricky. But yes, I think if anyone is in hiring panels and these things, I think what we should do, we should ask people for their open source projects, if they work on software like this, because I think it's really crucial that that our infrastructure is good, that our open source software is good. And yeah, so I would say it is a really cool career path. But yeah, it's never easy to find funding for these things, but absolutely crucial. So I hope that answered a little bit your question.
Yeah, and 10 years down the line, hopefully we don't come up with another paper about replication crisis in MRI, right?
Yeah, exactly. So we're trying to stay away from this, exactly. So no PNAS papers.
Yes. In the interest of time, I would like to invite our next speaker onto the stage. That will be Yongquan Ye from United Imaging in Houston, Texas. He'll be speaking to us about MulTiPlex MR, a reproducible multi-parametric qMRI solution. Yongquan, the stage is yours.
All right, thank you. So can you guys see the full screen?
Yeah.
All right, thank you very much for the kind introduction and good day, everyone. I am very happy to have this opportunity to introduce our latest multi-parametric MRI technique, which is called MulTiPlex. So the previous two speakers has talked about open data, the usage of open data sets that is already existing. So on my part, I would like to talk about the world technique that can contribute to this open data movement by providing a very good and reliable and reproducible data acquisition strategy. So I have to declare that I am the full-time employee of the United Imaging in Houston, Texas. So today we are going to have three parts. First, we do some background introduction, and then some technical aspects of the method, and then some applicability of this method. So when mentioned about multi-parametric MRI, a couple of names, a couple of techniques will pop into your mind, like fingerprinting, synthetic MRI, right? So the basic idea behind this multi-parametric MRI, I can sum it up as the 1 equals n and a bit greater than n. So this is meaning like we can use one single scan, we can acquire multiple signal contrast, and when combinedly and jointly evaluate them together, we can apply them in a much wider range of applications. So if we consider the multi-parametric MRI's contribution to the open MRI, so there will be some challenges. So I would say there will be challenges in the accessibility, applicability, and reducibility of these methods. So regarding the accessibility, these methods is easy to perform the data acquisition. Is it easy to implement? And when we got the data, is it easy to reconstruct and perform the post-processing? And regarding the applicability, this image of great quality, right? The resolution, the high resolution enough, 2D or 3D, is the scan time agreeable? Can we change the protocol and still get the robust results? And if we can use those analysis tools of the third party of our choice. And regarding the reproducibility, we would like the method to work across different scanning centers, scanners on different subjects, body parts, or diseases. So it will be a great challenge to have this multi-parametric MRI to contribute to an open MRI solution. So today we are going to introduce our method called the MulTiPlex, which was published last year. It is called multi-parametric MRI imaging with flexible design. So what can the MulTiPlex do? It can, with one scan of the MulTiPlex, which roughly takes about 3 to 7 minutes, it can offer you a 3D whole brain coverage with high resolution. and it offers you numerous contrasts and maps, which are strictly aligned with each other. So here we are showing some of the mappings, quantitative mappings, some of the contrast, and also it also supports the advanced process analysis of its own kind. So if we want to collect all these images using individual routine scans, it will generally take 40 to 60 minutes to do so. So it is a very fast and very good solution for a multi-contrast in MRI. So let's first take a look at some of the image results. Here are just some part of those images which are showing the major quantitative maps, the T1 maps, T2 star maps, photon density, and QSM, and also one AT1 weighted, which is a very strong T1 weighted contrast that you otherwise would have to acquire using an inversion recovery, but we did not use any. So with this scan, this is actually our routine brain scan at our 3T. The resolution is 0.67 by 0.67 by 1 without any interpolation. And the acquisition time is actually quite agreeable. It only takes like 6.5 minutes with 5 times acceleration with AI acceleration. So we can see that with this kind of high resolution, with one scan, we can already got all these images, all these mappings in a 3D display, and we can wield it with any within any imaging planes, and we can see all these results. They are strictly aligned with each other. And furthermore, those are just some of the maps. So beyond those maps, we can also get routine scans that we can use to match those that we get from individual routine scans, like the SWI or the true SWI. We can also generate double immersion recovery images like the gray matter only, white matter only, and CSF only. And we also mimic the T1 amperage. And we also have a very distinct photon density amperage that can show the white matter density, white matter lesions. And we can also use the immersion recovery to see the edge between the tissues, PSIR, or just perform the fast suppression. So with the multiplex, if we want to compare them with the routine scans, we can see that the first row are the images acquired with one multiplex scan, which only takes about 5 minutes and 6 seconds. We can immediately acquire the T1 map, the strong T1 weighted, density, T1 weighted, T2 star maps, SWI, and QSM. So if we want to, and using the same spatial resolution, and we collected the individual images using the respective routine scans, like the variable Philip angle for the T1 map, amperage for the PD weighted, T1 weighted, or the multi-echo SWI for the SWI and QSM. So we can see that when we're comparing the image quality, we can see that the multiplex images, they are of great quality, much higher resolution, and even better SNR than the individual scans because it collects a lot of data, as we'll see later. But the total scan time of all these routine scans, they sum up to roughly like 23 minutes. So we can, with the multiplex, we can acquire all this data with much shorter time and much better image quality. So how can we do that with this multiplex technique? So let's see about some of those technical aspects. In terms of the sequence design, here is the sequence diagram. So we can see that here are some of its features. So the base sequence of the multiplex is the 3D GIE. So when using 3D GIE, it's very good for performing a 3D high resolution imaging. And for the 3D, for the GIE sequence, it is very highly compatible with any acceleration techniques, from the parallel imaging to the compressed sensing all the way to the state-of-the-art AI accelerations. So any new acceleration technique can be readily applied to this multiplex technique. And also, the free DGRE is very easy to implement. And we use the dual flip angle to acquire the data. So with the lower flip angle, we got the photon density weighted. With the higher flip angle, we got the T1 weighted. And with a dual TR, we can acquire the B1 field information, which we can be used to correct for the T1 mappings. And we can see that we are using the multi-echo acquisition, and which give us a lot of those susceptibility informations we can use for the SWI, QSM, and T2 star. And optionally, we can also modify some of this acquisition strategies within each blocks of this acquisition blocks. So we can also got waterfall imaging or the MRA. So the multiplex has very big data, the very big and multidimensional data. So if we break down the multiplex data, we can see that because we have a dual flip angle, we have a single dimension along the flip angle. Similarly, we have the single dimension along the TR, along the echo times. And there's, because we are using the multi-channel array coils, so there is also a signal dimension along the channel dimension. So, when viewing with this, if we are using the 5 echo acquisition and 32 channel coils, the signal points for each channel we acquired will be as high as 384. So that means for each voxel we can acquire some very large sets of data for the calculations. So this also means the raw data size is very large. It can be anywhere between the 7 gigabytes to 100 gigabytes for one scan. So here comes another question. With such a big data, how can we perform the signal processing? So we have our own MDI signal calculation algorithm, which is called multidimensional integration. So this is the core algorithm of the multiplex. It calculates the major contrast like the 81 weighted, SWI, and the waterfall imaging, and all the major mappings. So here's how it works. If we are doing T2 star weighted, T2 star mapping, the routine procedure would first perform a color combination and then followed by fitting. And if we have multiple sets of data, it will average together. So, and then we get this T2 star map. Because the fitting process does not really work well for the low SNR regions like the background or the bones, we can see a lot of spikes in the T2 star map. But if for the MDI, we just simply input all the data at once into this function, and it will just directly generate the final T2 star map. And we can see that it produces a very low signal in the background and the bones. So many people ask me, are these images, are they of the same display window settings? Yes, they are actually of the same display window settings, and there are no background mask used on the MDI T2 star map. So the MDI method, if you are interested, please refer to our paper last year's. So here is the processing pipeline of the multiplex. The multiplex will generate 3 tiers of images, the tier 1 images and tier 2 images, which are the main set of the images in the form of the inline.com. So this will generate all those major image contrasts and quantitative mappings. And also a tier 3 images that can be calculated as a post-processing procedure. So for the tier 1 images, the original images that includes the magnitude images and the phase images of all the echoes. So they serve as the input for the tier 2 and tier 3 imaging calculation. And they also serve as a reliable means to determine the validity of the data. If we see any artifacts in the final results, we can come back and try to trace the source of those errors. This is actually very useful. So for the tier 2 images, we can offer the composited photon density weighted, T1 weighted, and T2 star weighted, which are simply calculated using the weighted average over all those echoes. So it offers very high SNR and high CNR. And there is a very new image contrast called augmented AT1 weighted, augmented T1 weighted or AT1 weighted. So generally, if we want to collect high, strong T1 weighted images, we will use immersion recovery methods like the MPRage. But if we already have T1 weighted images and photon density weighted images, and then we just divide them together, and actually we can obtain the immersion recovery light T1 contrast that is as strong as the routine T1 MPRage. So we can see that by dividing the T1 weighted images by the photon density weighted, we can get AT1 weighted images, and we can see that its contrast is almost the same as the routine MPRage. And for B1 mapping, which is used for the T1 mapping correction, can be simply calculated using the actual flip angle method. So it can provide a very fast and robust B1 field estimation with no additional B1 scan required. And there is the T1 mapping for generally for variable flip angle T1 mappings. It is generally done using the fitting along the flip angle directions. But because we are using double flip angle and double TR design in the sequence, it actually makes the problem becoming an analytic quadratic solution. So we can just simply solve this solution using the signal equations, and we can just directly calculate the T1 value using linear calculations. So we can see that because all the calculation involves all the echoes data, so we can see that the SNR of the T1 map is very high. And also the T2 star and R2 star mapping was the original method that we used in the MDI paper. So for using the MDI, we can directly calculate the T2 star and R2 star mapping with very high accuracy, very high SNR and efficiency. So the basic idea of using MDI to do this T2 star mapping is to calculate the signal ratio between the neighboring echoes and then extract the T2 star from this signal ratio. So we can see that using the MDI, the T2 star map and the R2 star map are of very high quality. Remember, this is a very high resolution imaging. So we do not see any spikes in the T2 star map here. And with the T1 and T2 star mappings, we can just directly estimate the photon density mapping from the original echo signals. So because by combining all the echoes results, the photon density weight mapping is also very accurate and also offers a very high SNR. And similarly, we can also use calculate the QSM using the multi-echo data. And finally, the tier 3 images are those virtual or combined contrast images that can calculate it based on the photon density maps, T1 maps and T2 star maps. So it's available via the dedicated MulTiPlex post-processing package, or very simple to do it yourself if you get a hands on this mappings DICOM data. So we can generate immersion recovery-like images like MPRage, stir, or the tissue boundaries, or we can generate the tissue selected imaging like the double immersion recoveries or the tissue probability maps. And also we can generate those combined contrasts, such as the true SWI, AT1 QSM, or the photon density flare. So finally, let's take a look at the applicability of the multiplex method. The most important thing as a multi-contrast MRI method and a quantitative method is its accuracy and robustness. So because multiplex is collecting a large sum of data during its scan, and we have the MDI algorithm for the signal processing. We are scanning the data with high resolution, and we got very high SNR by jointly combining all those data from different signal dimensions. So all these features come together, it will give a very good quantitative MRI results for the multiplex method. So we have tested our mappings on the system phantoms and on the health subjects. We can see that they are all very accurate and all very consistent between the different subjects and on the phantoms. And also the multiplex can be used on multi-body parts imaging, not only for the brain, but also for MSK or for the imaging in the pelvis. And depending on the body part, you can generate different types of the images accordingly. And multiplex is especially works better at ultra high fields with optimal settings because it's based on GRE. So it can the higher fields can give us a very higher SNR. And the GRE acquisition is very low SAR and PNS by itself. And by going with a high resolution, we can reduce the distortion in the images. So we can see that this is very good for the use on ultra high fields. And this is one of our best works. We have tested this multiplex in vitro rat brain on a 99.4 Tesla scanner. And we have acquired a 0.12 millimeter isotropic multiplex scan. So we can see that these images, they were all from the same scan. And we can see that this is a small rat brain that shows excellent details. And all the images, they are strictly aligned with each other. So with this type of high resolution, we have actually realized the first ever multi-parametric MRI imaging with the atlas levels of details. So there are, of course, many clinical applications all the way from the cancer imaging to stroke to the neurodegeneratives. That is not today's focused. So the final point to go back to our topic here is the openness of the multiplex methods. So in terms of accessibility, is the sequence, the multiplex is actually easy to implement for its data acquisition. Its sequence is actually quite easy to implement. It's just 3D GRE, but simply just add another TR to it. And the reconstruction, mostly of those in reconstruction is we see before is they are analytic and linearly. And we can also use some of those third-party toolbox for the QSM calculations. And for post-processing, there will be some vendor workstation, or we can just simply calculate it ourselves with the DICOM outputs. And because all the images are strictly aligned with each other, there's no need for any registrations. And in terms of applicability, it's acquiring the 3D high resolution data, which is the best form of the data. The quality has a very high SNR accuracy and repeatability, and also the robustness is very good. The scan time is definitely clinically agreeable with it 3 to 7 minutes to offer such a high resolution. And the protocol settings can be relatively flexible. So you can simply change the parameters and just click and scan, and you can get almost the same robust results. And for analysis, you can either use the vendor workstations or any third-party toolbox on the DICOMs. And finally, the reproducibility. Currently, the MulTiPlex is good for most of those motion-free scenarios, such as the head, right, the knee, the pelvis. If there is some movement like the liver, there could be further development to make it like a faster dynamic version of the MulTiPlex. It is also very ideal for the ultra high field use. So the field, it can be very compatible on all those different fields. And of course, because this is a new, relatively new methods, so it will be further clinically validated on its reproducibility and all these features for its openness. All right, thank you.
Thank you, Yongquan. I think the discussion will happen after the next speaker, as previously. So I'd like to now invite our final speaker of today's session, Julien Cohen-Adad from the Polytechnique Montreal in Canada, who will be speaking to us about quantitative spinal MRI open data.
Thank you very much for the invitation. There were a lot of excellent talks before me. It was a very interesting session. Okay, I think it should be working. All right.
Yep.
So hello, everyone. I'm going to talk about something lower than the brain, which is called the spinal cord. But essentially, it's MRI, and our goal is to just make the images better and share them publicly. So you all know what quantitative MRI is. It's an umbrella term for many techniques, which purpose is to look at the underlying microstructure of tissue in the central nervous system, such as myelin content and axon density. We can also probe neuronal activity using fMRI, look at metabolic concentrations, and ultimately all those quantitative MRI markers are valuable for helping the diagnosis and prognosis of trauma, various neurological disorders, and they are also useful as objective biomarkers for assessing the efficacy of drugs. While quantitative MRI has been widely used in the brain for more than 2 decades, it painfully makes its way to spinal cord applications. So why is that? One of the first challenges is related to the relatively small dimension of the image, of the structure to image. As you can see, the spinal cord is very small compared to the brain. It's about 1 centimeter diameter. It requires high spatial resolution, but higher resolution also means lower SNR. As you know, one solution is to get better hardware, including better RF coils, and also a more expensive solution is to get a more powerful magnet. Challenge number 2 concerns what is called susceptibility artifacts. So I'm going to illustrate this with this very nice tractography reconstruction that is overlaid on an anatomical image that is centered on the cervical spinal cord. As you can see, it looks like there is an interruption of the tracts around C6 vertebral level, which could be concluded as a complete spinal cord injury. In fact, I was the subject. I'm standing here in my office. So this is essentially a wrong interruption of the tracts. They should continue, and they stopped just because of a problem in the image, of susceptibility distortion. So there are ways to mitigate this artifact using faster EPI pulse sequences, other tricks, and another approach is to better shim to fix the problem at the source. So I'm not going to enter into these details, but it's just to give you an overview of what challenges we face when we want to image the spinal cord. Challenge number 3 So if you remember the slide I showed you at the beginning with the different quantitative MRI methods, in fact, I was only showing you some of the qMRI techniques. And every year at ISMRM conference, new sequences and qMRI techniques come up. So if a researcher is not familiar with qMRI, wants to start a project, what sequence and parameters should they choose? And in addition to choosing the right technique, there are also a lot of parameters to choose from in terms of SNR that they want to achieve, and what TR, what TE, and so on. So there is a lot of them to pick. So picking up the right method and parameters can be overwhelming. So that sets the context for what I'm going to talk about today. It's focusing on this challenge number 3, but addressing also the challenge 1 and 2, which is about a standard protocol. So the spinal cord imaging community has been working for the past 5 years, I would say, on a standardized acquisition protocol for spinal cord quantitative MRI data. The protocol is called the Spine Generic Protocol. 42 sites were involved, and importantly, it is available for the 3 main vendors using the product sequences. Study has recently been published in Nature Protocols, and the protocol is accessible at the address that you see in blue. An important note is that the protocol is not bound to a particular disease. It's meant to be as wide as possible in terms of applications, including MS, NMO, ALS, spinal cord injury, etc. Another important aspect is that the protocol is provided as a suggestion, so feel free to tweak it, change the timing, add more diffusion directions if you like, and so on. So what does the protocol consist of? Briefly, it includes a 3D T1 weighted scan that includes the whole brain, cervical and part of the thoracic cord, a higher resolution T2 scan for cord atrophy measures, a gradient echo, multi-echo 2D axial scan to measure white and gray matter. And the gray matter measurements could be useful to look at atrophy in ALS, for example. And the protocol also includes a diffusion-weighted sequence to compute myelin-sensitive metrics and a magnetization transfer protocol to look at myelin-sensitive metrics as well. So on this axial view, there is an overlay of 4 spinal tracts that are of general interest, including the descending corticospinal tract and the dorsal column. So a particularly important aspect of this protocol is that it includes a comprehensive document with step-by-step procedures that is directed towards the MR technicians. So it's ideally someone looking at the protocol should be able to reproduce the images across different sites. We like to look at images. So here are 3 images across 3 vendors acquired on the same subject. I was the subject, so I was careful to not move too much. I knew this images would end up on a slide on a presentation. That's for a T2 weighted image. And the different contrasts now showed in axial orientation for the T1, T2 weighted, T2 star, MT without and with the pulse, T1 weighted, and the diffusion weighted scan. So usually in conference and articles, we always show the good-looking data, but I also want to show you the bad-looking data with a lot of awful artifacts. So here are some examples of an MP RAGE taken in the same participant, but at 2 different sites. And you see that the slightly larger lordosis on the left induces a more pronounced CSF, a cerebrospinal fluid flow and CSF motion resulting in an artifact that prevents to nicely delineate the spinal cord. Another artifact likely due to a poorly recovered CSF combined with flow effects on a T2 weighted scan, an axial view of a multi-echo GRE scan with patient motion and without patient motion, respectively top and bottom. That's a funky looking artifact. It's actually a sagittal view of a diffusion weighted image. Okay, but it has been acquired with a sequential acquisition as opposed to ascending or descending. And there was motion in between the odd and the even lines, which results in this very difficult to correct image. Poor fat saturation and also poor shimming, and this is due to pulsatile effect on the diffusion weighted scan, which is highly sensitive to bulk motion, right? These are MR tech mistakes with a wrong coil selection, for example, or swapping the empty on and empty off. Also changing the shim settings. So all those things are annoying, and ideally we would like to look at those issues once we gather all the scans from the different sites. So speaking of which, this standardized protocols was also accompanied with two publicly available datasets. The first one is a single subject who was acquired across 19 different sites. And the second dataset consists of 267 subjects acquired across 43 sites. So it's not the 267 subjects who traveled, it's just that there was approximately 6 to 7 subjects per site. And then they were aggregated into this big open access database. It's organized into the BIDS structure, and as Steffen mentioned, it's also leveraging the Git annex and DataLad infrastructure for accessing them. So briefly, the way we organized this data set was that each site acquired and anonymized the data, then sent it to the managing team. The managing team did some additional curation. We launched some PyBits validator to make sure that the data were compliant. We looked for artifacts and so on. And when we're happy, we pushed those data on an AWS S3 protocol server, and the metadata are still hosted on GitHub. So as Steffen nicely described, in order to access the data, it's very easy. It's a single command line with Git annex or with DataLad, and you could download those data sets. So our lab is a strong advocate for transparent and reproducible science. So there is a website of the project where you can find the description of the data analysis, how to run it, how to perform the quality control, and the manual corrections of the intermediate results. I'm gonna show you some results of this multi-subject database. So these are the sagittal T1 weighted scans for all the subjects. So you can already see that across the 3 vendors and across the 42 different sites, the FOV placement was well respected. So we're quite happy with that. The analysis was fully automated, done with Spinal Cord Toolbox, and the first metric of interest, which is particularly useful in neurodegenerative diseases such as MS, is to look at the cord cross-sectional area at C2, C3 vertebral levels, because it's a good indicator of the severity of some disease. Like looking at the atrophy of the cord is a good metric. So what we see in black, blue, and green are the 3 different vendors, and within each vendor, there are also different models. For example, the Siemens, which has the majority of the data set, there are the Prisma, PrismaFit, Skyra, Trio, Vario scanners. And the little dots represent one participant. Okay, so when we compute the intra-site, so for each site, we compute the coefficient of variation. This coefficient of variation was below 8%. And within Vander, the inter-site variability was below 4.2%. So if you want to look at, a cross-sectional area on Siemens scanners, you can be, confident that the COV will be below 4.2%. Another metric, quantitative metric, is the MTR, magnetization transfer ratio, which was computed in the white matter, an average between C2 and C5 levels. The inter-site COVs for all vendors were below 2.5%, so an even better reproducibility for this metric. There are two crosses here that you can see, and those crosses are there because the acquisition parameters were different than the one prescribed by the protocol, so they were excluded from the statistics. And lastly, results from the diffusion-weighted scans using the diffusion tensor model, where FA was computed and shown here. Again, coefficient of variation, inter-site and intra-vendor were below 4% for all vendors. So we are pretty happy with this result. There are more results, which I don't have the time to show here, but again, they are available online. So if you type the spine generic protocol on Google, you can click on the website and you can navigate it to have more information about the data set. You can actually interact with the different views of all the contrasts from different sites. And if you go into the analysis pipeline section under the results, you'll actually see the tables, the plots that I just showed you, but you can interact with them. You can actually find the actual values for the cross-sectional area from the T1 weighted scan, but you also have access to all the other metrics. So to conclude, the spinal cord generic protocol promotes replicability, dissemination of knowledge. It facilitates the addition of spinal cord qMRI protocols at the non-expert centers. It minimizes variability in multi-site, multi-vendor studies, and it's already implemented in at least 50 clinical sites and also used in wide multi-center initiatives, including the INSPIRED study and the CanProCo study, which is a pan-Canadian study, which is led by Jihan Ho at University of Toronto, and it concerns MS patients. The open access spinal cord data that I showed you could be used for creating normative values that could then be compared with patients, and could also be used for improving models, like deep learning models for for segmentation of normal structures. So we hope that this recent paradigm shift in acquisition and analysis of spinal cord MRI will hopefully pave the way towards a more systematic inclusion of spinal cord in studies. But there's one remaining aspect which we have not talked about, and it concerns communication, and in particular discussions about unmet needs between physicists, clinicians, and MRI vendors is key. And in order to solve that problem, we've been organizing spinal cord workshops since 2014 every year, except during COVID. And these workshops are a great opportunity to meet and discuss bleeding edge methods and to push the field towards a better implementation and adoption of some of the qMR techniques. So if you'd like to be included, please visit the spinalcordmri.org website, and you can subscribe to the mailing list. With that, I would like to acknowledge everybody in the lab who contributed to these studies, the collaborators, and the funding sources, and thank you very much for your attention.
Thank you, Julien, for an excellent presentation. I mean, a personal thanks from my end as well. I've learned a lot from the open science and the transparent philosophy that your lab follows. So I really appreciate it. I think now it's the discussion section. So hi, Yongquan. So there are already a couple of questions in chat for you. So congratulations, very impressive work. Sophie asks, I might have missed it, but do you use deep learning in any pipeline, either reconstruction or synthesis of different contrasts?
Yeah, thank you very much. For the reconstruction pipeline, we actually have a minimum use of the deep learning because actually we introduced the MDI algorithm, the multidimensional integration. So actually that one, because I show a lot of slides and did not went into details on the MDI method. But actually the MDI method is one of the most important feature, if not the most important feature of the multiplex. So it actually calculates perform the calculation of those mappings and all those most important contrast images using the complex data. So they are all computed using the complex data, and also making good use of the channels, the multiple channels of data. So we do not first combine those channels to give you one of those combined images in the magnitude or the phase. We actually just process those channel data in its original form, which is complex and which is individual. So by using this, we make good use of the Gaussian distribution in the noise and make them cancel each other during the MDI calculation process. So if you remember that when I showed the T2 star mappings results, comparing between the MDI methods and the routine fitting methods, we can see that those backgrounds, the routine fitting methods have a lot of spikes in the backgrounds, and MDI have basically 0 spikes and very low signals in the final T2 star map for the backgrounds. That's because all those Gaussian feature of the noise was used. So and the MDI calculation is pretty fast. It's very fast. So in terms of the reconstruction pipeline, actually we do not have, we just simply do not need to use too much deep learning algorithms.
Maybe a follow-up to that. So you did show a slide comparing conventional acquisitions to the reconstruction of multiplex, right? And if I remember correctly, the multiplex reconstructions already include some form of B1 correction. So is this done online? Because typically if you acquire all these data sets, then you would need to do all these things offline.
Yes, they are online. Actually, it is the WIP sequence that is implemented in the United scanners. So yeah. So for all those tier 2 images, so if you remember, there's big chunks of images called tier 2. All those tier 2 images are to be reconstructed in all those major contrasts.
Yeah, I think the next question also synergizes with the question that I had. So it's a very powerful sequence that you have and the post-processing, but how does it compare to multi-echo MP2 range or other MR fingerprinting like sequences, which also provide you multiple quantitative contrasts?
Yeah, that's a very good question.
From other vendors, right? Yeah.
Yes, I think it's from Siemens, right? The MT2 range also, if I remember correctly, also offers T1, T2 star, and QSM. But I don't think it offers the photon density mapping. And the most important feature of it is it is inversion recovery based. So that means it has a lot of dead time, and its data acquisition is not very efficient. So if I remember correctly, its position is very long, and there's not much way to, accelerate it, because you have to wait for the emotion recovery to, to go back to the equilibrium to give you a good signal. And also because it has to collect the data very quickly during the recovery of the signal. So it will use very high bandwidth. So that would also reduce the SNR of its signals.
Yeah, and one comment that you had in one of your slides was motion is or could be a problem, because it's one scan that gives you all these contrasts. So if there's motion, then it affects all the scans. Are there like approaches like prospective motion correction or something that could be employed together with this technique to make it better, you think?
Yes, of course. Motion would always be a problem when you perform any 3D high resolution scan, right? So so that's why we are using a lot of acceleration. So we are using for deep learning, we actually use the deep learning for the acceleration. So that's up to like 6 to 8 times acceleration if we go with high enough resolution. So that brings the time down to 3 to 7 minutes. And if there is still motion within this time, that it will just look like the ghosting artifact, just we see in
Blurring,
any other 3D, and the blurring, just as we see in any other 3D methods.
Yeah, thank you. Yeah, I see a comment in chat that Julien's already answered, but for everyone watching on YouTube, Steffen asks, do you see a good use case for QSM in the spine, Julien?
Yeah, I do see a lot of good use cases, but hard to apply. So yeah, I mean, I know a couple of groups working on it. Where I'm kind of involved in a QSM project in a pediatric spinal cord. It's very, very difficult. Hopefully, by fixing the problem of shimming, we might be able to get reasonable, QSM results.
I had a question related to acquisition. So with the brain data across multiple sites and vendors, the gradient nonlinearity can be quite a problem. And at least with the brain data, I do see that near the neck and the spine is where it's more pronounced. So is it similar with spinal cord acquisitions? How big of a problem do you think it is?
Right. So I mean, yeah, I mean, especially for large field of view, the gradient nonlinearities become a problem. But I mean, if they could be reasonably well corrected with the gradient specific like matrix that comes on the scanner. It's usually done on the console. It could be done offline. I know we've played with the UK Biobank, and there was a talk about the UK Biobank data. We did play with because the problem with the UK Biobank is that actually the processed data cropped to the spinal cord. We wanted to look at the spinal cord, right? So we had to go back to the source data, which are not gradient nonlinearity corrected, and then we had to hunt for the grad coef file, and then apply it to the thousands of that it was funny. But once you correct for this, it's usually reasonably correct in terms of like spatial correction. Yeah.
And regarding the P1 in homogeneity issues and things, do you think there are applications for like these universal pulses and other PTX approaches or on the spine? Like, it's a very naive question from my end.
I'm wondering if you're one of the reviewers at ISMRM. We actually submitted a universal pulse for 70 in the spinal cord. So if it's accepted, then you'll know more in a couple of months.
Very nice, keeping the suspense. So there's a question from Jan. Can you comment on the importance of dynamic shimming in the spinal cord?
The importance of dynamic shimming. Dynamic meaning like a slice-wise, I assume slice-wise B0 shimming. It is actually very helpful. I mean, there's very, very good work done by Jürgen Finsterbusch and colleague, Volk Eipert, Merv Kaplan, for fMRI application. So when you want to get proper axial slices with the GRE EPI in the spinal cord for fMRI, that's where dynamic shimming could be very useful. So, and I guess moving to 7 Tesla, it will be even more interesting. So we are pretty like actively involved into improving dynamic shimming in the spinal cord for 70 applications. Notably with another open source software called Shimming Toolbox, which was recently out in MRM. But yeah, I think this is a very beneficial technique. Yeah.
I do not see many more questions in the chat, but if I may ask a more general question, so you did show a lot of data that goes across sites and countries and probably dealing with multiple REPs and whatnot. So I always have this problem that the question of anonymize is like the definition, it's very unclear. So was there any specific issue with sharing of all this data that you encountered across these many sites?
Yeah. So was the question for me or?
Yeah, it was for you, Julien. Sorry.
Yeah, it was a bit tricky, but I thought it would be actually more, more tricky than that. But at the end, each site dealt with their own, IRBs. So I was not so much involved. I had to sign a few paperwork for some of the countries, but it was relatively smooth. Yeah. I mean, this project is a long process. I think it took us like 5 years to get to the end. So there is definitely things to improve from the like the legal aspects, but that's a bigger discussion.
Yeah. No, it was just a throwaway question at the end. I think we are on time, even though we started a bit late. I would like to thank all our speakers for such inspiring presentations. And thank you all for listening on YouTube and those who are live here at the moment. Yeah.
Thank you very much.
Thank you very much.
Take care.
Take care. Bye-bye.